Welcome to Unit 5, Lecture Video 1. So Unit 5 is actually broken down into 3 chapters. We're going to do Chapter 10, then we're going to do Chapter 8, and then Chapter 9. So we're going to start off with Chapter 10. So the first part of Unit 5 is Chapter 10, which is about DNA, RNA, protein synthesis, and so on and so forth. Remember, there are 4 main classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. We touched a little bit on proteins last chapter when we discussed enzymes. Okay, we're going to talk a little bit more about proteins in a couple of lectures when we talk specifically about how proteins are made. This lecture we're focusing on the nucleic acids. So the nucleic acids, there are 2 of them. It's DNA and RNA. And remember, these guys are basically your blueprints. They're your genetic information. So the whole purpose of it is to have a blueprint for your body and then to also be able to pass that blueprint on. There was this really pivotal experiment called the Hershey-Chase experiment, because those were the scientists that did it, Hershey and Chase. But what it basically did is it actually showed us that DNA was the genetic material. At the time, we thought we could pass DNA onto our offspring, or we could pass proteins onto our offspring, and we weren't sure which. So this experiment basically had 2 setups. Other than that, they're exactly identical. Here is where it differs. Radioactive. So if you remember way back when, okay, radioactivity is basically it's a physical condition. The biggest thing is that we can trace it. Okay, if you've had an MRI, all right, that's radioactivity that we can actually trace. We can basically just measure it, and so it's really helpful. So in one experiment, so in the top here, they made the protein radioactive to see what happened, and in the bottom here, they made the DNA radioactive and to see what happened. So through the course of this experiment, in here, the radioactivity didn't get transmitted into the new cells. So it stayed in the liquid, whereas here the radioactivity did get transferred into the new cells. And so that basically showed us that DNA is the genetic material. And we've had many experiments since then that have helped back this up. So what is DNA or RNA? So these guys are nucleic acids, so deoxyribonucleic acid and ribonucleic acid, that are basically made up of a 3-part monomer, okay? And that 3-part monomer is called a nucleotide. So our 3 parts, 1, 2, 3. A sugar, when we're talking about DNA, it's deoxyribose. When we're talking about RNA, it's ribose. Our phosphate group, our nitrogenous base, okay? And then there are 5 nitrogenous bases. 1, 2, 3, 4, 5. So we have adenine, guanine, thymine or uracil, and cytosine. So what they are, as you can see here, we've got the phosphate group, which is there. We've got the sugar, which is there, and then the nitrogenous base, which is there. So that's what makes up the nucleotide. And one of the things that we figured out is that DNA and RNA both go through what's called a complementary base pairing rule. You can see that down here. You may remember talking about this in middle school. It means that if we have an A, it's going to be partnered with a T if it's in DNA, or a U if we're talking about RNA. And then if we have a G, it's going to be partnered with a C. And so they match up. So if you notice, we've got 1 purine, adenine, with 1 pyrimidine, thymine or uracil. 1 purine, guanine, partnered with a cytosine. Okay, and so what happens is they actually then match up, and this is what the rest of this chapter for the most part is going to be based on. So it's really important for you to understand this base pairing rule. So what happens is if we have an A, we're going to have a T on the other side. If we're going to have a G, we're going to have a C on the other side, or a U in the case of RNA. DNA and RNA, like I said, are both nucleic acids, and they have a lot of similarities, but a couple of differences. Number one, DNA has to, for the most part, stays inside the nucleus. It doesn't go anywhere. It's the control center. It stays there. It doesn't move. Sugar is deoxyribose. Remember that OSE ending means it's a sugar. Thymine, okay, that is the base. Okay, we have thymine. Double-stranded and anti-parallel, which we're going to talk a little bit more in the next lecture when we talk about how it is replicated. Sorry, I had to think of that word for a second. Okay, and then this is the actual genetic material that is transferred from generation to generation. This is what we give to our offspring. This is what you were given when you were born. Okay, genetic material that, and then your cells, take that genetic material and turn it into your RNA and your proteins. Okay, RNA can leave the nucleus. Ribose sugar, uracil, usually is single-stranded, but it can be double-stranded. And then this is basically just the intermediary that carries out DNA's instructions. So DNA can't leave the nucleus. So what it does is it makes RNA basically do its dirty work for it, gives RNA some instructions, sends RNA out, and then RNA does its job. Okay? RNA is made and recycled and made and recycled. Okay? So here we go. You can see, here's a picture. All right. Here, it's that double helix that you guys are used to seeing. And here you can see we've got two on either side. Here we just have one. Okay, and then it basically we have it looks like a ladder that we've twisted, and here it's just one side of it. So this does different things. So, and you can see, so when I was talking about purines and pyrimidines, all right, here you can see the purines. All right, and then we have our pyrimidines. And if you notice, if we add them up, okay, because we always have a purine with a pyrimidine, we get 1, 2, 3 rings. Okay, 1, 2, 3, because remember they're going across there. All right, same thing on this side. Okay. So here's your wrap up. How did phage T2 help us understand DNA? And what is the complementary DNA strand to the following sequence? So we're just talking about DNA. So what's the complementary DNA strand? ATG, CA, TTG. Okay, this was a short one. I'll see you in class. Have a great day.